people going, welcome this evening. My name is Brad Bold. I'm part of the biology department, and what's going on tonight is actually the 29th annual, and we still haven't come up with a better name for it, but 29th annual Science Night. And for some people, they go, oh man, that doesn't sound good, but if you're here, then you know it's a good thing. And actually, this part of it is really linked to what we now call the annual Science Teachers Night Out, because we every year for the last 29 years invited local science teachers, and very pleased to have them come, give them a meal. You're not getting it. We provide a meal and spend a little time talking and just getting to know each other. And I'm really pleased. I'm not going to run off all the names, but representatives from Cambridge Middle School, Mora High School, North Branch High School, Cambridge SNE High School, St. Francis Middle School, Hinkley High School, Ogilvy High School, and Chisago Lake. So really pleased that for those of you folks that are here representing your schools, we really appreciate you coming. For the rest of you, and I know many of you are students and you have to be here, no, play along with me, okay? Like you really want, yes, that's the answer. Yeah. Oh, jacked up on something. Anyway, this is part of really our 50th anniversary. Well, it's the 50th anniversary of Anoka Ramsey Community College, and Cambridge is one of the campuses, but we as a campus are really 37 years old. She's coming with the hook. Okay, anyway, so there's lots going on. Lots going on both campuses, and really celebrating the fact that we have been here for many years providing hopefully good instruction. That's our intent. Anyway, I just want to invite you to a number of things going on campus here, and then I'm going to introduce that lady there. Anyway, what's coming up Friday night is the Scholar Dazzle Scholarship Fundraiser, and our foundation just does an amazing job of generating funds for scholarships and maybe some of you students are recipients, and I hope that's the case. But anyway, that is Friday night, and it is, where is it? Is it Spirit River Community Center. For those of you that know, it's old spectacular events, but good times, and it's going to be a fundraiser. What else is coming up? Where's Mary? Mary, are you here? Mary's not here. She's always doing something. But Mary Januszka came up with a great idea, I think it's about 14 years ago, of having a candlelight ski here. So this Saturday evening, if you are free, man, come on back, because we're going to have about 400 flaming bags. Well, I think they're little luminaries down along the river. There are trails there. It's beautiful, and the weather's going to be good. And if you're interested in cross-country skiing, you can cross-country ski. If you want to just walk the trails, you're welcome to. There are a bunch of prizes I'm going to be giving out. Yoo-hoo! Honestly, there's a bunch of prizes. There'll be hot chocolate. There's going to be music. If you know, the music started. If you know Kenny Crona and who's with them? Rick Steiner. They're going to be here. Great musicians. It'll be a good time. And it's free. Price is right. What else is happening? Starting January 30th, which is Saturday, going through March 4th, we have the privilege of exhibiting art by a local artist, Fred Anderson. And I'm not going to give you all the information on that, but he's a great local painter, and his work is going to be exhibited here during that time. What else, you say? Could there be more? Yes. Every year, I don't know how many years this goes, and some of you are going, it's years now. But every year we have a Minnesota writer, and I'm not familiar with this gentleman, but R. Vincent Moulise. I don't know if I pronounced that right. I'm guessing not. But he's a poet, and that is going to be Thursday, February 18th, right here in this room, and the event is free and open to the public. All of these things, if you're really interested, the information will be online, and it's online right now, but apparently he has won numerous awards in finds agency, humor, poetry, in the adventure of being an urban Indian. So some great diversity sort of information. And, we have a play coming up here. Lisa, we were practicing just a moment ago, and that's coming up on February 25th through the 27th. I'm almost done. Earth Friendly Fair, April 30th, and it's a great time, loads of activities for the kids, and the Environmental Club. Anybody in the Environmental Club here? Okay, Environmental Club, you do a bunch of great things. You have, there are vendors, there's fire, I don't know. But
Also have a health fair. The nursing students are putting on a health fair that same day.
I was going to get to that, except no, I didn't have it written down. Thank you. There's going to be a health fair that same day, and part of the 50th anniversary. I don't know how the words run and fun come together, but they do. 50th anniversary, there's going to be a 5K fun run. 5K, I think that is, those are in like hundreds. So 500 miles. All right, I'll quit. But thank you so much for being here. And normally I would just introduce the speaker, and you'll find out why I'm not going to. I'm going to introduce Sean Meyer, who is one of the nursing faculty on campus here, and you're going, why not just skip to the speaker? Oh man, you're going to find out in just a second.
How about I just talk? It all started one day when I was making copies in the copy room, and Brad said to me, as we had just had our cancer poster presentations in the nursing department, he said, do you know of anyone who might like to speak? And about science night, and I thought of my sister. Okay, so I will introduce to you Monica Akre. She's giving me a hard time because I don't know all where she's been teaching. Okay, so she's taken several career detours before arriving on her current path of graduate student in molecular genetics. She has taught at Winona High School biology, anatomy, physiology, in Rochester 7th grade life science, in Seattle at a community college, and she helped there with cellular biology. For 6 years, she also worked at Mayo Clinic's cytogenetic laboratory. She was then encouraged by doctors there to transition to research and go to grad school. She's in her 4th year at the U of M, studies drug responsiveness in multiple myeloma. She has published twice, soon to do it again, and then she hopes to finish her doctorate within the year. And so, Monica Akre. Okay, good idea.
So then when I was 12, we had it all set up, trust me, but we're almost home, we're almost there.
Keep going.
I've got nothing. Wait.
Tell him a joke.
I do animal calls.
Okay.
We're going to resort to that. Any requests? Cow. Okay. Our president is here for the first time ever, and he's going, oh man.
No, it's fine. Okay. Oh, there's still a lot. It still won't work. I think you tell me.
I know, I see
okay.
But I think it's on.
The video mute is on.
Yeah.
Okay. All right. Well, greetings, everyone, and thank you so much for coming out tonight. And I'm super excited to talk to you about science, life science in particular. So we're going to narrow it down. Tonight is Science Night, and we're going to talk about life science. We're going to talk about biology. And so I was wondering what I should talk about tonight. And a couple of different topics came up, and I decided to whittle it down to life and death. Just, so I hope you brought provisions for the next several days so that we can get through this together. But I'm actually just kidding. I'm only going to talk for about 2 hours, an hour. So what I want to talk to you tonight about is the power that is wielded within each and every single cell of a living organism, specifically you. So, let's begin. The average human body is composed of cells, tiny, oil-bound bubbles, lots and lots of them. Most recent estimates are that there are 37.2 trillion cells in a human body. I love how they add that .2 in, as if that's going to really wow you, but that is the most accurate estimate as of now. They used to estimate it based on weight or volume, but those were inaccurate because cells are different weights and volumes. So this is the most recent updated estimate, and that is that you are made up of 37.2 trillion cells, and I hope that wows you. And if not, we're going to get more deep. So each of these cells right now is extremely busy, building down and building up and tearing down proteins, growing and dividing, and yet others are more relaxed and in sort of a meditative state of sorts, with very little change going on. But I want you to understand your power. So in order to do this, we're going to do a little time travel exercise, magic school bus, if you will, back to your beginnings. And you maybe never wanted to think about this before, but let's. I'm going to make you. I am, for the sake of brevity and perhaps the dignity of your parents, going to skip past the part where the twinkle in the eye happens and go straight to what I call the Great Sperm Race. So, this is a high-powered image of that great sperm race and what that time looked like when you were an egg waiting for a sperm. Now, as you can see, this is the egg, and there are many, many, many sperm, all diligently and urgently working to deliver important messages to that one waiting cell. This is a brutal race. And these sperm only have this little tiny flagella with which to whip around with and to try and escape the acidic environment of the vagina as they try and get to the egg. So already these sperm are winners per se because the majority of the sperm didn't even make it this far. But in this great sperm race there are no awards for second place. So, sorry guys. There's only one winner. So, as soon as one of these sperm digest and infiltrate the cotton candy-like coating of the egg, there's a sudden transformation in the egg, and suddenly its membrane is impervious, like steel, to all of the other efforts of the sperm. They cannot get through. So just imagine this. You were seconds away from being infiltrated by a totally different sperm. Each sperm has a shuffled variation of genetic material, and you would not be you 2 seconds later, 3 seconds later, 4 seconds later. Somebody else would have won. So I want you already in your seats to feel like winners. You won the great sperm race. Yeah. So good job, you. So after the sperm releases its very special delivery of genetic material, this is what it looks like. So this is a fertilized egg, and these two bubbles right here are pronuclei, which contain, for the human, usually 23 chromosomes each, and they merge together, making 46 chromosomes. So that's a lot, 46 chromosomes, but and I just wanted a little disclaimer here, instructions equals DNA. Sometimes I toggle back and forth between those two terms. So each one of these chromosomes, one from your mom, one from your dad, if you stretch it out, it is amazing how much you can pack into one of these little guys. In fact, each one of those pronuclei contain 1 meter worth of DNA, each of them, which means that the cell in total has 2 meters worth of DNA. So I just wanted you to get a real picture of this. So I'm going to show you a real life experiment of this idea, who is probably right now really regretting that she consented to these pictures. So, small disclaimer for scientific accuracy, this is not the exact event whereby my daughter initially received her DNA from me and my husband. Nonetheless, it's a touching image to impress upon you the incredible amount of DNA held within each cell. 1 meter from me and 1 meter from her dad, in every single cell, microscopic cells. Those molecules of DNA are wrapped tight. Amazing! And we're not done yet. Are you feeling great? Great sperm race, wounds DNA. Yeah, we're making progress. So let's do a little math. You have 1 fertilized egg, 2 meters worth of DNA, and in that DNA are recipes, genes for making proteins. Now, I like to deal with analogies and allegories to help me understand the microscopic because it gets incredibly dull occasionally. So let's just imagine that the cell, with all of its DNA, is a baker, a very happy baker, with a ton of recipes. And in this case, we are talking about 50,000 different recipes. Some of the recipes are copied absolutely verbatim. Some of them delete items, leave stuff out. Some of them have to alter the recipe a little bit. But in total, if your cell was a baker, they would be able to make 50,000 different kinds of things. That's also amazing. So you can imagine that if the cell was an individual, they would be extremely busy. Sometimes they would make the same recipe every single day, and sometimes they would have never seen a recipe before. They would never make it. And so if we're to take that analogy to our cell, some genes are never made into proteins. Some genes are made constantly. So this is important to get across for where we're going next, because we're still time traveling. You're still a single cell. So let's go forward and remember about you being fertilized. And what happens next is the cell copies every single recipe, all 2 meters worth of DNA, so that when it divides into 2 cells, it can give a copy to each cell. When that division happens, and this is very important, it is unequal. What do I mean by unequal? Here's a cell, and the protein concentrations down here are not the same as the protein concentrations up here. So when the cell splits and divides, there is a discrepancy between this cell and this cell. They are different. That's really important. If they were the same, you wouldn't exist. So in development, you have Division, change. Division again, change. Division again, change. Each time an unequal distribution of protein concentration. Those proteins are what directs the new cell to make different proteins. So different. That's the big deal here, is that things are getting different. So right now, we've had a lot of division, we've had a lot of changes, every cell is different, but they're clinging together as a ball. And what happens next is somewhat unnerving. This ball of cells needs to get food fast. It's hungry. So it excretes proteins that gently digest away the lining of your mother's uterus. You actually got under her skin. That's not just a phrase. You burrowed in there so that you could get nutrition, blood, oxygen, remove waste. And so at this point, we're going to go and speed up this simulation a little bit so that I can impress upon you what these different cells and their differences, because I really tried to drive that home, what that means for the growing embryo. Your brain started out as a tube. A tube. But the cells were different along its length. And the cells at the top of the tube, the anterior portion, grew faster. And then they migrated. They dove down and then came back up, rising to form what you know to be your cerebrum. Or maybe you don't know it, but know it. It's your cerebrum. And it started out as a tube. And the only reason why those cells did what they did is because they were different from the cells behind them and behind them. That is what generated the signals for your brain to develop as it did. I think that's amazing. I'll give you another example. Okay, so I want you to take your hand and make a fist. So your hand, as it was developing, looked something a little like this, like a paddle. And this is a mouse, but you look the same back then, maybe less green. But it started out as a paddle. Now I want you to imagine the groupings of cells on each knuckle having a difference that inspired them to grow more quickly, to outpace their neighbors in the soft spaces in between your knuckles. And extend your fingers as they grow and grow. And as they grow, the cells in between, from their very own DNA, are instructed to die. This death, this dying, this signal is extremely important to the emerging shape of your hand. And the cells in between yield to it. That's what's being shown right here in the in the boxed area. These cells are dying back. This cell death happens all the time. It's very precise, calculated, necessary. Believe me, if you're upset about your size now, if you did not have any of your cells die, you would be much larger. So it's a necessary thing. And when these cells die, they do it in a very tidy, dignified manner, whereby they break up and fragment into little tiny bubbles that are digested and absorbed by the surrounding cells and used for nutrients for them. So in that way, you can believe in reincarnation of the cell. It is born again into the neighboring cells. Use whatever imagery helps you out. And so the dance goes on, and cells sink into their identities as fingers, skin, muscle, bone. They incorporate other tissue types, cell types, so that they may become organs and function as one. So just from one single cell, the human form emerges. Such diversity exists from the same 2 meters of DNA. Your liver cells, your brain cells, your skin cells, all your cells came from one cell. How is this possible? How is it possible that the same instructions gave rise to such diversity? And it's all in what recipes you've made, what genes are expressed, what proteins are made. So that's pretty powerful. That's the life part. Now, the next part of my talk is a little bit more grave. With this great power that we have in each cell comes great responsibility. So what happens when genes are made out of turn, or recipes are mistakenly made or changed or mutated? Then what? Well, usually nothing. We've all made mistakes in the kitchen, right? And usually, you just throw it in the garbage, it's all good. And that's what happens to your cells, too. Mistakes are made, and there are no severe repercussions. And yet, occasionally, when cells have damaged DNA, or when they make aberrant proteins, there are consequences for the neighboring cells and for the organism as a whole. This we know as cancer. Now, what is cancer exactly? In short, it's a grouping of cells that take on certain characteristics because of the DNA and changes in the protein concentrations that allow them to grow faster than their neighbors. They forget their boundaries and start to invade the surrounding tissues. Then, the cancer we usually talk about here is the kind that takes away the functions of other organs. So, the primary tumor, the collection of cells that first starts growing and dividing more quickly, is usually not detrimental to the organism as a whole. What do I mean by that? Well, if you have a lump from breast cancer, you have a lump in your breast. It doesn't impede any other organ functioning, and you can live. So why is cancer a problem? Because after this initial proliferation, it can carry on and acquire more mutations and more changes. And what it can do then is that it can invade the surrounding tissues and go into the bloodstream, a highway to the rest of the body. Most of the cells that are able to do this and break off from the original tumor will never survive. But unfortunately and occasionally, some do. They arrive at a new organ, take up residence, and start to form a secondary tumor, what we know as metastasis. And this is a huge problem in cancer. In fact, it's what causes 90% of cancer deaths. It's not the primary tumor, it's the metastasis. It crowds out its surrounding neighbors and disrupts the organ where it landed so that it can no longer function. So it's worth noting that these cells that are able to initiate a metastasis and go to another organ undergo an extreme transformation. For those of you who are interested, it's the epithelial to mesenchymal transition. I see some people writing down, so I feel like I should say something that's wordy. So these cells actually revert back to a more embryonic, immature state. And that's what makes them so pernicious and able to adapt to a new surrounding, is that they revert back to a more embryonic state. So, cancer is really hard to treat because when we attack cancer, we attack ourselves. Cancer comes from us. It is us. But it has characteristics, a personality that is unique to it due to the excessive proteins it mistakenly makes or the proteins it neglects to make. If we identify which proteins exactly are being over or undermade, we can formulate a regimen specifically targeted to the cancer and not the normal cells. We could find the Achilles heel. And this is where I get to my thesis work. We can do this. We are doing this. There are huge data banks publicly available to you tonight when you go home. Download them and see if you can figure it out. Patient A, the red spots indicate proteins that are being overmade. The green spots indicate proteins that are being undermade. And that is specific and unique to this patient. That is the story of this patient's cancer. Out of their 50,000 different proteins, this is what made their cells cancer. Patient B overexpresses and overmakes the red proteins in a different way than patient A, and undermakes the green proteins. She will not respond to his treatment, and he will not respond to hers. However, we know that the pattern of expression directly correlates with what treatments will be effective. This is called personalized medicine, and we are just learning more about it. Right now, it's in the stages of being treated on a gene by gene by gene basis. We know that certain patients who have certain mutations and certain genes won't respond to certain treatments. That is, I'm sorry, it's pedestrian. We need to understand what this patient's whole cancer is telling us about how it can be treated. Not just one spot, and perhaps not just one drug. Indeed, we know that regimens, combinations are more effective than single treatments for the majority of cancers. There are exceptions. So what I am doing is taking a look at these expression patterns and taking a look at the responsiveness to different drugs and trying to figure out which of these proteins will tell me if this treatment is effective or will not help at all. So, I wanted to show you something else that I feel is an honor and a privilege to get to work with every day, and that is cell lines. Some of you may be familiar with the story of Henrietta Lacks, also an HE. The HeLa cells, yes, no? Okay. That is just but one cell line. There are hundreds. Specimens donated, usually from cancer patients, that can be propagated in the laboratory. We put fake blood on them, a cocktail of different mixtures, different chemicals, and we coax them to grow. And they do. And they outlive their hosts. This is a cell line that came from a colorectal cancer patient. This cell line has outlived its donor. And the interesting thing about these cell lines is that they grow in culture and they also can be manipulated. I can take these cells and take DNA that I suspect causes the cell to die, and I can package it in a little oil bubble and put it in its food, and it will merge with the cell, and the cell will welcome it into its genomic DNA. And then I get to see whether or not it makes an impact, whether or not it does indeed cause the cell to die. And in this way, we can start to understand the complexities of cancer and all of the networks that are impacted by it. Sometimes we do this on a gene-by-gene basis, and sometimes we do it with a whole series, a whole array of genes. But I do want to get back to the work that I was doing with the gene expression. That was all done on cell lines, 600 different cell lines from patient donors from different cancers. And we have expression data for every single cell line. We also have data on how those cell lines responded to 100,000 different drugs and natural byproducts. If you're interested, it's called, it's on the Sanger website, and it's also done in conjunction with the Mass General Hospital, and it's free for the taking, which right now that's a huge initiative that cancer needs all hands on deck in order to understand it and to figure out the patterns and the puzzles that it presents. So I am very thankful to all of the donors who allow me to work on these cells every day and how they have pushed the field forward. And so I just wanted to, again, thank you so much for letting me come here and talk to you about my passions in life and death. And I will take any questions. Yes, that did not take long.
Is the data from those cell lines, is that available, like, right on a current basis to doctors who are treating cancer?
See, the issue with that is, yes, it is available. Do they know how to decode it? They do not. So there needs to be more preliminary work done. So also the individual patients, when you take a sample and you do an expression profile on that, we don't know how to match it up quite. We don't know what the Achilles heel is yet. What is that pattern of red and green and yellow that will tell us that it's going to respond to this drug or not? That cartoon was the hope of what we can do in the future. But I will say that those publicly available databases are available on Excel. You can download it tonight and find out which drugs are responsive or which cell lines are responsive to a drug and which cell lines are resistant.
Where do we find this information?
Okay, so I'm going to mess up the acronym, but it's called the Genomics of Sensitivity to Drugs in Cancer. I got it right. And it's part of the Cancer Genome Project. So if you type in that and Sanger Institute, it should take you to the website. Yes, in the back.
I have a little more technical question. Was that microarray? I'm sorry.
I have a little more technical question. So those gene expression profiles were with microarray data?
Yes.
And if they were, have they been validated with RNA-seq data? Okay. And the other question is, do you have any primary patient samples and not cell lines? Because the more you grow them, the more they change in culture, right?
Right, exactly. So she brings up a couple good points. First of all, so as we progress through science, we get new technologies that are amazing, that make the old technologies kind of obsolete. So in this cancer project, we're kind of at the BCR stage, and she wants us to be at the DBR stage. So expression, or seq is better than expression arrays, is the point. And no, they're all arrayed, Affymetrix arrays. The other point that you brought up that was a good one is primary cell lines versus primary samples versus cell lines. And this is a debate that is common in conference rooms at the U of M all the time. You're going to get this debate. Cell lines have been taken out of their host and have been grown and multiplied, propagated for many, many, many generations. So how like the original cells are they? There can be changes along the way. Is it giving us information that's really going to be relevant in a patient? Or is it just sending us on a wild goose chase? The ideal would be to experiment on humans, which is unethical. But we start with cell lines. Once we have good reason to believe that those cell lines have given us valuable information about drug therapies, we move to an animal model. Once we prove safety and efficacy within an animal model, we go to clinical trials. Or you mentioned primary patient samples, where you can take the samples out of the patient and grow them for a short amount of time in culture. But it is a race against time, because when you take the cancer samples out, they may or may not survive, and your data may or may not be useful. So it is I don't mean to say, oh, it's just as easy as, tonight, download this file and cure cancer by tomorrow morning, okay? It's not that easy. But with the cancer cell lines and with 600 different cell lines, we're more confident than we would be with 50 cell lines per se.
Do we know how many different mutations can cause cancer?
Oh, wow. Okay. So, no, we don't. And that's a great question. So, there are driver mutations that sort of, like the engine of the Titanic, drive a tumor down into becoming more carcinogenic or progress oncogenically. And then there are passenger mutations that, are mutated, but they really don't have a role in the characteristics that make cancer. So we are having trouble figuring out which are the passenger mutations and which are the driver mutations, especially because everybody's cancer is different. The reasons why they get cancer, what promoted the cancer, what initiated the cancer, it's all different, so it's really hard to tease out. So that is the job of bioinformaticists, to try and figure out what caused the cancer and what didn't. And that's still very debated. How many genes cause cancer? We're trying to figure that out.
You sent me an article about how biopsies can cause cancer to spread, and it was about a vacuum biopsy. Can you talk more about that?
Well, first I sent you 2 articles, I think. Hopefully you read both of them, because I tried to send you one sort of pro and one sort of con. But for a long time, and so most of you know how needle biopsies work with, let's say, breast cancer. Okay? So they stick a coring needle into the tumor, guided by radiology, and they take it out. And within that core, kind of like an ice core, they have the cells of the tumor. Now you can imagine that that's kind of not only an invasive procedure, not as invasive as, say, a mastectomy, but when you go in and when you go out, you are traveling past blood vessels. And what is the image that I just showed you? It was of cells of the tumor being broken off and going into the blood vessels. And what we've learned from reading articles that your sister sends you about that, is that even though many, many cells from the tumor are dislodged during a needle biopsy, they have no evidence that it causes metastasis. Why would that be? It is because in order to survive in the bloodstream, you need to have a certain concentration of proteins. You have to be ready, fortified. It's like going on a camping expedition in the wilderness. You don't know where you're going to land, you don't know what you're going to need, and you may die along the way. No dissuasion to going to the area. So what happens when you take a needle biopsy, those cells are disrupted, but we don't have any evidence of any of them reaching the blood vessel that are prepared to take on that challenge and to colonize another organ. So that was an issue, and they created a vacuum biopsy to try and get rid of that risk. Then they found that the risk wasn't there. Somebody probably was disappointed.
I guess I would have a question for students here that are taking notes. You've written some things down and you're looking at them and you have no idea what you wrote. You've been in my class. But the reason that I say this is that Sometimes you sit and you write things down and you don't know what they are. You have to ask. So are there some things that you have on your notes right now that you really like some clarification on? Because here's the person that can answer it.
Absolutely, and I feel as though if you don't understand something on your notes, I have done you a disservice. So please let me clarify for you. I would hate for you to go out without knowing.
Is there another animal species that has the comparable ratio that we do to cancer?
You know, somebody asked me a similar question in the teacher's night right before, and I don't know. We talked about lifespan and how this I do know. So I'll say something about this, and maybe I can get away with answering your question. And that is this: the single variable that determines whether or not you're going to get cancer isn't whether or not you smoke, isn't what you eat, isn't your body size. It is your age. The older you are, the more likely you are to get cancer. Exponentially. So, in the case of animals, they have shorter lifespans than we do, as someone pointed out. So, it would follow that they wouldn't have the same rate of cancer as we do. But, to be honest, I don't know.
Go. Get back first and then no.
Could this even get more complex? Like, do you talk to like theoretical physicists and stuff that get into, say, string theory? Could that be the unseen elements that even could be involved in all this process?
I don't even know how to answer that question. So, could it be more complicated? It's fairly complicated right now, and my guess is that we could use a little help from all disciplines. We use we employ some computational biologists who work on computers and algorithms and mathematicians, and so could we welcome some theoretical physicists into the mix? I'm sure we could, as long as they bring donuts. I'm not sure what they would have to contribute, but if I did know what they had to contribute, we might be collaborating with them right now. I'm not sure.
So you're saying computers cause cancer? Is that what I'm hearing?
Yes.
Okay, I'm gonna be honest with you. I came because I'm in Sean's class, and I'm like, oh, this sounds really cool, because cancer, right? Cancer kills people. There's all sorts of different kinds of cancer, but I didn't know much about it. But my question for you is, I know some people who have had cancer and have, died from it, and each one of them went to these support groups, and a major topic that they talk about in those support groups are this whole idea of, opium oils, that kind of thing. Is there, I'm sure it's developing right along with the pharmacology testing everything you can, but is there any solid research that there's anything to do with cannabis oil or whatever that could actually impact the cells? And if it does, is it like one uniform, this is the stamp all cure? Because if it is, we should probably be using that. Just a thought.
Yeah, if it is, we probably should be all using it. And to my knowledge, those 100,000 compounds that I talked about, I'm not sure that is included in it. However, I know that there are natural byproducts that are included in the screen. What we did in our screen, just because it's really hard to make a nice little matrix with 100,000 compounds, is we used the ones that are focused on multiple myeloma treatment right now, and the ones that are FDA approved, because then we can give it to patients as soon as we find an efficacy for it. So, in my research, we have not touched upon that, but nature has given us a lot of amazing things. And so is it possible that nature could be effective in treating cancer? I don't doubt it. Have we found it yet? I'm not sure. It could be in that screen and we just haven't figured it out yet.
So why is that such a relevant topic at these, say, support groups and such? Are there people who are just clinging to that as an idealism? Or is this something that maybe there's other research being done?
So as far as the research is concerned, because that's what I can comment on, the research that's being done is publicly available. If there's research being done outside of that, I'm not aware of it. And as far as what a cancer patient clings to as they struggle, I cannot speak to that, and each personal struggle is different. So that is a very valid and different topic that I am not certified to bring to bear.
There are quite a few doing research on cannabis, though, dude. I'm just saying.
Okay, so you said the older you are, the more likely you are to get cancer. And why is that? What about elderly people makes them more susceptible to that? Is it that they've been exposed more longer than the younger people to things, or?
That's a good point. Okay, so there's a couple things that I want to say, and that is, firstly, we understand that exposure to viruses has an impact on cancer development, and that is either because the cells that have the virus are excreting elements that promote cancer, or it's because the virus actually integrates into the genomic DNA and disrupts normal protein production. So those are two things, and as you age, you get exposed to more viral components. But also, so as a cell progresses and marches on from that first cell, every cell division, it gets exposure to things that could damage the DNA. Every cell division, it replicates the DNA, and the possibility for mistake is there. Though there are molecules, a multitude of molecules that stand sentinel to repair any mistakes that do happen, mistakes do go through and go forth. So there's a couple things that are in play as far as age contributing to cancer. It's not as though it's a cold that they catch more easily as they age. It's because of damage incurred over a lifetime. Things like a sneaker cell, literally, that has been quiet and well behaved, but just needs one more insult before it becomes cancer. Does that answer your question the way you wanted? Okay.
Get my exercise. I'll get right to you. We have one back.
I missed your introduction, so I apologize if you already mentioned this, but you said that you, in the bank of information, you tested multiple myeloma drugs. And they were all solid tumors where you used those drugs because they were FDA approval. They were all non-solid tumors, hematological cancers.
So if I'm understanding your question correctly, we are using the database and pulling out B-cell malignancies, including multiple myeloma, and using FDA-approved drugs that are already FDA-approved for blood-borne cancers, or for blood cancers, hematologic malignancies. So we're not taking the cell lines that are from solid tumors to determine whether or not the drugs would be effective. However, there is out there the idea that it doesn't matter the cancer that you come from. You could illuminate a lot by being tissue of origin blind and taking a look at the expression profiles and understanding what cancer is.
And I agree with that. So,
yes,
so here's my next question
then. Initiative.
It makes sense. So my next question then is, when you do those screens, are you really looking for, with molecular inhibitors, again, That is my phone, I'm sorry. Against mutations in particular molecules, or it's just you guys go like pharmaceutical companies go and throw whatever and see what works, and right? Because the idea is that you're hitting the same genes mutated in different tumors, doesn't matter what their region is.
Right, right. So as far as the database that I referred to is concerned, they threw every compound, I think they just cleaned the shelves. They threw every compound that they possibly could on different little cell dishes containing cells from 600 different cancers. And they wanted to see the response. They didn't need to know that, oh, this was the pathway, because let's be honest, we don't know how a lot of drugs work. Do we need to know how the drug works in order to know that the drug works? So right now there's an urgency to just get the treatments out there, because the treatments available right now are chemotherapies, which are pretty toxic and lower the quality of life for the patient, and that's unacceptable.
One thing when you're going through it, and I was wondering if students would ask, is sometimes we get caught up in using jargon that we assume. You threw around the word, oh, no, I've forgotten it. No, but what was it? Just not the profile, but
Array?
The array. Can you, because I'm guessing somebody's out here going, I don't know what an array is. So
Okay.
Can you just define that, please?
So let's use a mnemonic, sort of. An array is like a rainfall onto a slide. It's a rainfall of different regions of the DNA, those 2 meters, and it's slotted down in discrete little spots on the slide, and it tells you whether or not those regions are expressed or not. And when I say expressed, that means did the baker make that recipe? So when I say array, I mean a whole bunch of different genes at the same time. Originally, when we studied cancer, we studied like one gene at a time. We looked for the Philadelphia chromosome or a MYC mutation. We studied one gene at a time, and now we're doing it array-wise, where we look for all of them at the same time. So that's an array.
I work as a counselor here at the campus, and I have students that come in and talk about careers. Sometimes students will say, I don't want to be a nurse, I don't want to be a doctor, but boy, I'm sure interested in sciences. I don't want to be a dentist. What kind of courses, what kind of options, what kind of recommendations would you have for a beginning student who is just very curious and interested in science, and they like technology, they're on top of every little gadget and exploratory technology that comes out. Do you have any recommendations that we could share with younger or people who are beginning their careers?
Uncertifiable advice. So take the courses that you love to find out what you love. And then what you do with it, if you want to go into research, try to find yourself in a lab, because that is going to give you unique skills to that lab. I think about where I work and the lab that I'm in, and I walk 2 feet this way, and somebody's working on something totally different, on something that I am unfamiliar with. So there are a wide set of skills that you couldn't possibly learn in one class, 2 classes, 3 classes. Find out what you love, get the basic skills, and then get yourself into a lab if this is what you want to do.
And as much as it pains me to say it, on this campus we really haven't had opportunity, or maybe it's student numbers or whatever, but I have to mention Paula, who was asking the questions back there from the Coon Rapids campus. They have really a wonderful undergraduate research I don't know how you want to say it, project, classes now that are being offered. So as much as I would hate to see any of you leave this campus, to be honest, I do want you to understand that Anoka Ramsey offers great opportunity in undergraduate research. And I can't even give all of the right credit here, but Paula is one of the instructors, and there are several others. And if you're interested, maybe Paula can hang around, or certainly you can get connected. But only if you really want to do undergraduate research. Otherwise, I highly recommend microbiology this summer on this campus. We've got a... Other questions? We'll have to wrap it up pretty soon here, but if you have a couple more questions, wow, this is good stuff. More animal sound? No. Well, one more question.
So I'm cheating. I did my PhD also in Van Ness lab in multiple myeloma, so I want to tell you, I'm gonna ask you. Sorry. But what is, it always fascinated me how a terminally differentiated cell can become a tumor. Okay? So these are plasma cells, they're antibody secreting cells that they de-differentiate and become tumors when they actually have learned their full function already, which normally doesn't happen. So what's your theory, right, on how this deprogram, because the way I understand it is, you know what I mean? I mean, the way I understand it is you don't learn your function, you divide, and that's why you look mesenchyme-like, okay?
Right.
But plasma cells learn how to do their thing, and all of a sudden they accumulate enough mutations to go wacko, they produce antibody that is not functional, right? So
here's the dealio. I spend some philosophical moments in tissue culture, feeding my cells and looking at them under the microscope. It's a lot of brain time, and sometimes I talk to the cells. And I just wonder to myself, how did you get here? What's your story? Why couldn't you just make antibodies like everybody else and just die and be done with it? And so a couple of things about plasma cells. These are the blood cells that come down the lineage. You know, you have the stem cells, and then you make all sorts of different cells from that. These plasma cells are specific in that they make antibodies to antigens. And in order to do this, let me back up. As a baby, you get sick really easily because you have not built up your immunity. You don't have a repertoire of plasma cells that make antibodies to all of these different antigens that you have seen. How do you build up that repertoire? Well, there is a gene sequence that is kind of dangerous. It is prone to shuffling and switching and changing and rearranging in order to make different shaped antibodies in the hopes that it will touch on one that's invading the body. So you have genes in your body that are programmed to mutate, to change, to rearrange. That's a little bit dangerous, and it's kept under tight control in almost every cell type except the plasma cells, because their jobs is to make antibodies. So when you have programmed mutation and shuffling, it's dangerous business. Also, as plasma cells, you are able to hang out longer and de-differentiate in order to continue on and give the your host immunity for future attacks. So those two things together, the ability to escape programmed cell death and the ability to mutate, If you combine those together, you can have a really deathly combination. And so it's my hypothesis that multiple myeloma co-opts those and takes it further than it was licensed to do. That's what I got.
So somatic hypermutation, but it doesn't happen with leukemias, right?
No,
Or
just with myelomas.
So how about lymphomas? They do somatic hypermutation, but they aren't fully differentiated.
That's a totally different ball of wax from another paper that is in submission right now about the Apovex, but that's for a different time.
Some of my past students recognized random somatic gene recombination.
That's what she just said. You're going, what? Any other questions?
Any other questions? No, really, any other questions?
In the past year, I've come across a few articles that have gotten quite a bit of recognition in the media about pathogenic treatments or cures. What are your thoughts on these positive or negative?
Oh, okay, so like reprogramming T cells in order to attack.
Or even bacteria or viruses that are programmed to attack.
Yeah, so that's so what he's talking about is can we use pathogens to attack cancer? Can we, almost like a bloodhound, give them the scent of the cancer and say, go to it, boy. And that would be a wonderful therapy, wonderful. I don't know much about it, but there are people at the U that are investigating it. With each cancer, you would have to get that particular scent. So that is problematic, but it's a great, tidy little therapy that leaves the rest of the cells alone while attacking only the cancer cells. So that's interesting, promising.
Is there potential for them to go haywire, maybe Rambo style?
One could imagine. Yes?
I saw some kind of TV show, not a news story, where they were talking about that, and they had treated somebody. They genetically modified the virus or bacteria so that it wasn't infectious in that way, like it couldn't cause disease, but it could still infect the cancer cell and kill them. I don't remember specifically which one it was.
That was a Walking Dead.
It was modified.
Yep. So they have adenoviruses that are non-infectious, and so they can do gene therapy and gene editing that way. And that is also under investigation. I think you were talking about the cellular therapies where they train the T cells to attack the antigens of the specific tumor. Viral therapy and gene editing is different and more controlled.
All right, thank you so much. And once again, if you're around this weekend, please come out on Saturday for the candlelight ski. It's really a great time. It's a family time. Hopefully see you next year. If you have a topic for us, let me know.
I'm really happy we weren't talking the whole time, huh?